Welcome to EPG Pathshala. I am Aparna, working as scientist in MFPI Food Quality Control Laboratory, Professor Jayashankar, Telangana State Agricultural University, Hyderabad. Today, we are going to learn about water-soluble vitamins. In this, we will see what are the different kinds of water-soluble vitamins, properties of the water-soluble vitamins, solubility, and modes of degradation of water-soluble vitamins during the processing. We have already seen what are the vitamins in the previous chapter, that is fat-soluble vitamins. Let us now understand what are water-soluble vitamins. In water-soluble vitamins, we have vitamin B1, it is also called as thiamine. Then we have vitamin B2, which is also known as riboflavin. Then we have vitamin B6, which is known as pyridoxine. We have vitamin B12, which is also called as cyanocobalamin. Then we have niacin, it is also called as nicotinic acid, and pantothenic acid, folic acid, biotin, and vitamin C. So vitamin B1, B2, B6, B12, niacin, pantothenic acid, folic acid, biotin, and vitamin C will be discussed in this topic. The first vitamin is vitamin B1, which is also called as thiamine. Thiamine is very widely distributed in most of the living tissues of both plants as well as animals. In most animal products, it occurs in the form of phosphorylated form, whereas in the plant products, it is present in the form of non-phosphorylated form. Commercially also, thiamine is available either as thiamine hydrochloride or thiamine mononitrate. Both these salts have specific areas of application and their uses depend on the product matrices in which they are used. A considerable amount of research has been carried out on heat stability of thiamine as well as its salts, particularly in the context of cooking losses of thiamine. Early work on thiamine losses during bread making showed an initial cleavage of thiamine to pyrimidine and thiazole. The destruction of vitamin by heat is more rapid in alkaline media than in acidic medium. Vitamin B1, usually present at the rate of 0.04 milligrams of thiamine per 100 ml, is usually lost during various types of processing. The losses are 10% for pasteurized milk, 5 to 15% for UHT milk, and between 30 to 40% for sterilized milk. You can see 30 to 50% B1 activity losses in the production of evaporated milk or condensed milk, which implies that higher the amount of treatment or processing technology, higher is the loss of vitamin B1 or thiamine. Losses of thiamine during the commercial baking of white bread are anywhere between 15 to 20%. Part of this loss is due to yeast fermentation, which can convert thiamine to co-carboxylase, which is less stable than thiamine. Thiamine is very sensitive to sulfides and bisulfides and is cleaved by the sulfite. The reaction is rapid in high pH and causes larger loss of vitamin in vegetables which are blanched with sulfides and in meat products such as comminuted meats where sulfides and bisulfides are used as preserving agents. When the pH is low, such as in citrus fruit juices, etc., the bisulfides occurs mainly as unionized acid and the thiamine loss in such systems are not very significant from those in the normal ones which contain bisulfite. Research also has shown that the rate of sulfite-induced cleavage on thiamine during preparation and storage of minced meat shows that losses of thiamine were linear with sulfur dioxide concentration up to 0.1%. The storage temperature did not have a significant effect on the loss of thiamine. It has also been reported that thiamine is cleaved by aromatic aldehydes. Thiamine is decomposed by both oxidizing and reducing agents present in the food matrix. If it is allowed to stand in alkaline solution in air, it is oxidized to disulfide and small amounts of thiazolone. A range of food ingredients also have been shown to have an effect on the stability of thymine. In general, proteins are protective of vitamin, particularly food proteins such as egg albumin and casein. When heated with glucose, either as a dry mixture or in a solution, a browning analog to a Maillard reaction can occur. This reaction is similar to the reaction between sugars and amino acids and may be important in loss of thymine during heat processing. Thymine is unstable in alkaline solutions and becomes increasingly unstable as the pH increases. The stability of vitamin in low pH solutions such as fortified fruit juices or drinks or beverages is very good. The stability of thymine is adversely affected by presence of copper ions in some kinds of food matrices and food processing techniques. This effect can be reduced by addition of metal chelating compounds such as calcium disodium EDTA. The heavy metals only appear to influence the thymine stability when they are capable of forming complex anions with constituents of the medium. The enzymes thyminases, which are present in small concentrations in a number of animal and vegetable food sources, can also degrade thymine. These enzymes are mostly commonly found in a range of seafoods, for example, shrimps, clams, and raw fish, but are also found in some varieties of beans, mustard seed, and rice polishings. Two types of thyminases are known and are designated as thyminase 1 and thyminase 2. The former catalyzes the decomposition of thymine by a base exchange reaction involving a nucleophilic displacement of methylene group of pyrimidine moiety. A problem associated with addition of B1 to food products is the unpleasant flavor and odor of the thymine salts. The breakdown of thymine, particularly during heating, may give rise to off flavors and off odors and different kinds of compounds which are derived by degradation of vitamins, which are believed to contribute to the cooked flavors in a number of foods. However, both thymine hydrochloride and mononitrate are relatively stable to atmospheric oxygen in absence of light and moisture, and both are normally considered to be very stable when used in dry products with light as well as moisture-proof packaging materials. After understanding thymine and the loss of thymine during various processing techniques, let us move forward to the next water-soluble vitamin, that is vitamin B2, which is also known as riboflavin. Riboflavin is most widely distributed of all the vitamins and it is found in all the plant and animal cells, although there are relatively few foods which are rich sources of riboflavin. It is present naturally in foods in two bound forms. One is riboflavin mononucleotide and flavin adenine dinucleotide. Plants and many bacteria can synthesize riboflavin and it is also found in dietary amounts in the dairy products. Riboflavin is available commercially as a crystalline powder that is only sparingly soluble in water. As a result, the sodium salt of riboflavin 5-phosphate, which is more soluble in water, is used for liquid preparations. The most important factor which influences the stability of riboflavin is light, with the greatest effect being caused by light in 420 to 560 nanometers range. Fluorescent light is less harmful than direct sunlight, but products in transparent packages can be affected by strip lighting in retail outlets. Riboflavin and riboflavin phosphate are both stable to heat and atmospheric oxygen, particularly in acid media. In this respect, riboflavin is regarded as being one of the most stable water-soluble vitamins. It is degraded by reducing agents and becomes increasingly unstable with increasing pH. While riboflavin is stable in heat processing of milk, one of the main causes of loss in milk and milk products is due to exposure of the products or milk to light. Liquid milk exposed to light can lead to loss of 20 to 80% of its riboflavin content in just 2 hours. The rate and extent of loss can depend upon the light intensity, the temperature, the surface area of the container exposed. Although B2 is sensitive to light, particularly in a liquid medium like milk, it remains stable in white bread which is wrapped in transparent packaging and it is kept in a retail outlet or in any kind of store. After understanding riboflavin, let us now move forward to the third water-soluble vitamin, that is niacin. In nature, pantothenic acid is widely distributed in plants and animals but is rarely found in free state as it forms part of coenzyme A molecule. It is found in yeast and egg yolk and also in the muscle, tissue, liver, kidney and heart of animals. It is also found in a number of vegetables, cereals and nuts. Pantothenic acid is optically active and its dextrorotatory forms have vitamin activity. Losses of pantothenic acid during preparation and cooking of foods are normally not very large. Milk generally loses up to 10% during processing. Meat losses during cooking are not excessive when compared to those of other B vitamins like thiamine or riboflavin. Free pantothenic acid is an unstable and very hygroscopic oil. Commercial preparations are normally provided as calcium salts or sodium salts. The alcohol form panthiol is available as a stable liquid but it is not widely used in the foods. The three commercial forms, calcium and sodium dipantothenate and dipantothenol, are moderately stable to atmospheric oxygen as well as light when protected from the moisture. All three compounds are hygroscopic, especially sodium pantothenate. Then we will move forward to the next water-soluble vitamin, that is folic acid or folates. Folic acid does not occur in nature but can be produced commercially. The naturally occurring forms are a number of derivatives collectively known as folates or folacin, which contain one or more linked molecules of glutamic acid. Polyglutamates predominate in fresh food, but on storage, these can slowly break down into monoglutamates and oxidize to less biologically stable or available folates. The folic acid synthesized for food fortification contains only one glutamic acid. The stability of folates in foods during processing and storage is variable. Folic acid loss during the pasteurization of milk is normally less than 5%. Losses in the region of 20% can occur during the UHT treatment and about 30% loss is found after sterilization. UHT milk stored for 3 months can lose over 50% of folic acid. The extra heat treatment involved in boiling the pasteurized milk can decrease the folic acid content by 20%. Losses of around 10% are found in boiled eggs, while other forms of cooking like frying, poaching, scrambling, etc. can lead to 30 to 35% loss. Total folic acid losses from vegetables as a result of heating and cooking processes also can be very high. Let us now go to the next vitamin, that is vitamin B6. It is also called as pyridoxine. Vitamin B6 activity is shown by three compounds in form of pyridoxal, pyridoxal and pyridoxamine, and these are often considered together as pyridoxine. Vitamin B6 is found in red meat, liver, cod roe, milk and green leafy vegetables. The commercial form normally used for food fortification is the salt, which is pyridoxine hydrochloride. Pyridoxine is normally stable to atmospheric oxygen as well as heat. Decomposition is catalyzed by metal ions. Pyridoxine is sensitive to light, particularly in neutral and alkaline solutions. One of the main causes of loss of vitamin in milk is sunlight, with almost 21% loss being reported after 8 hours of exposure. Pyridoxine is stable in milk during pasteurization, but about 20% can be lost during sterilization. Losses during UHD processing is around 27%. Average losses as a result of roasting or grilling of meat is about 20%, with higher losses in stewed and boiled meat ranging from 30 to 60%. Cooking or canning of vegetables also can result in vitamin B6 loss up to 20 to 40%. After understanding vitamin B6, let us now move forward to cyanocobalamin, which is also called as vitamin B12. The most important compound in vitamin B12 activity is cyanocobalamin. This has a complicated chemical structure and it occurs only in animal tissue and as a metabolite of certain microorganisms. The other compounds showing this vitamin activity differ only slightly from cyanocobalamin structure. Vitamin B12 is commercially available as crystalline cyanocobalamin, which is a dark red colored powder. As human requirement of vitamin B12 is very less to almost 1 to 2 milligrams per day per person, it is often supplied as a standardized dilution on a carrier. Cyanocobalamin is decomposed by both oxidizing and reducing agents. It is sensitive to light and ultraviolet radiation also. Vitamin B12 is normally stable during pasteurization of milk, but up to 20% can be lost during sterilization and losses up to 25 to 30% can occur during the spray drying of milk. The stability of vitamin B12 is significantly influenced by the presence of other vitamins also in the same food matrix. We will go to the next vitamin, that is biotin. The chemical structure of biotin is such that 8 different isomers are possible, and of these only the dextrorotatory or D-biotin possesses vitamin activity. D-biotin is widely distributed but in small concentration in animal as well as in the plant tissues. It can occur both in the free state, in milk, fruit and some vegetables. It is also available in bound form to protein in the animal tissues and yeast. It is also commercially available as white crystalline powder. Biotin is generally regarded as having very good stability and being fairly stable in air, heat and daylight. It can gradually be decomposed by ultraviolet radiation. Biotin in aqueous solutions are relatively stable if the solutions are either weakly acidic or weakly alkaline. Avidin, which is a protein complex which is found in raw egg white, can react with biotin and bind it in such a way that biotin becomes inactivated. Avidin can be denatured by heat and biotin inactivation does not occur when eggs are cooked and consumed. Then among the last water-soluble vitamin, we have vitamin C. Vitamin C is widely distributed in nature and it can occur at relatively high levels in some fresh fruits and vegetables, and it is also found in some animal organs like liver and kidney. Small amounts can be found in milk and other meat products. The L-ascorbic acid which is present in the foods is easily oxidized to dehydro-L-ascorbic acid. In fresh foods, the reduced form normally predominates, but processing, storage and cooking increases the proportions of dehydro form. Commercially, vitamin C is available as L-ascorbic acid and its calcium, sodium and magnesium salts. It is also available as ascorbyl palmitate and can be used in this form as an antioxidant in different kinds of processed foods. Ascorbic acid and ascorbates are relatively stable in dry air but are unstable in the presence of moisture. Ascorbic acid is readily oxidized in aqueous solutions. Conversion to dehydroascorbic acid is reversible, but the products of later stages of oxidation are irreversible. Destruction of vitamin C during processing or cooking of foods can be quite considerable, with losses during pasteurization being around 25%, sterilization about 60% and up to 100% in ultra-high temperature stored milk which is stored for 3 months. Milk boiled from pasteurization can also show losses between 30 and 70%. Losses of vitamin C also are found after cooking and hot storage of vegetables and fruits. The commercial dehydration of potatoes also can lead to loss of vitamin C between 35 and 45%. Destruction of vitamin C during processing of vegetables depends on physical processing which is used and the surface area of the product which is exposed to oxygen. Slicing and dicing of vegetables will also increase the rate of vitamin C loss. Blanching of cabbage and other vegetables can also produce losses of up to 20% of vitamin C losses, while subsequent dehydration can lead to another 30% loss in the vitamin C content. Let us understand the vitamin-vitamin interactions, which is one of the least expected and least understood aspects of maintaining the stability of vitamins. This can lead to more rapid degradation of one or more vitamins in any kind of food or a beverage. And these interactions should be taken into consideration when vitamins are used to restore or fortify products which are present in the liquid phase, such as in soft drinks or fruit juices. Most of the work related to vitamin-vitamin interactions has been carried out only in the pharmaceutical industry in relation to the development of liquid multivitamin preparations. 4 of the 13 vitamins have been identified as having interactions with each other with deleterious effects. These are ascorbic acid, thiamine, riboflavin, and vitamin B12. So it is important to understand the vitamin-vitamin interaction so as to maintain the stability of majority of the vitamins which are present in the food matrices or fortified to the food matrices. It is very important to understand the addition of nutrients to the foods also. There are various methods of addition. The first one is restoration. It is nothing but addition to restore the original concentration of key nutrients. Then fortification, which is known as addition of nutrients in amounts significant enough to render the food a good food to superior food, which is a source of added nutrient. Then we have the third category. It is nothing but addition of specified amount of selected nutrient in accordance with the standard of identity or legal permissible amounts. Then we have the next one that is called as nutrification, which is a generic term which is intended to encompass any addition of nutrient to the food. So we have learnt about the water-soluble vitamins. All the water-soluble vitamins, their stability, their losses during the processing, and the method to maintain the integrity and stability of the water-soluble vitamins in the food matrices. For further information on this topic, you can refer to Food Chemistry textbook by Nelson, and you can also refer to Advanced Food Science textbook or any other standard textbook to understand more and in depth about the topic. Thank you.